Perfect. And so thanks so much for sticking around. I know it's been a long day. I'm sure some people are fatigued. Everybody can relax for this talk. I was given the nice and gentle title of talking about therapeutic and advances in research in young onset. And actually, do you know what? 10 years ago, that would have taken about 5-10 minutes. It probably would have been a fairly handy talk to go through, but actually so much is going on in this area at the moment, and we've touched on so much of it throughout our earlier talks that I really am not going to be able to even cover a fraction of it. So what I was trying to think what's most useful, and I think what's most useful is to maybe think of what is the stuff that people ask us about. They read in the newspapers, they're wondering where are we at with regards to this, where are we at with regards to the other thing, and oftentimes it may be them talking about themselves or maybe talking about their relatives. Just because I'm going to be talking about therapies, I have done consultancy work with some of the companies that we'll talk about later on. This has really been gone through, so I'm not going to labour it. We've talked about the variety and the heterogeneity, the differences that we see in young onset dementias versus maybe the majority of dementia being attributable to Alzheimer's disease after the age of 65. We've also talked about how people with young onset dementia are disproportionately affected from the point of view of the delays in diagnosis, from the point of view of stigma that's attached to it, and from the socioeconomic burden. And really, I think a key thing, and we didn't actually talk to each other really about our presentations, but a key theme that has been running around all of this is the importance of diagnosis. And when you sit in a clinical room and you talk to somebody who's receiving awful news, and their overwhelming sense oftentimes is relief that somebody has finally put an actual label on it and has said with a degree of confidence this is what it is, you really do get to appreciate the importance of a diagnosis for where people, you've got to have a starting point that you have a certainty in to be able to navigate and map around the rest of it. So it really is that most important of things, and I've just kind of put up this slide, I'm happy to share these slides, is there are these validated. So, for any of us that are working in memory services, or who are interacting with people in memory services, we have to know these are the scaffolds by which we direct the consultation. So one of the things we haven't probably dived deep in today, and actually was Joan's word, Joan earlier on who used that phrase, dive deep. And it's actually around, you've got to know this scaffold to be able to ask the questions, to be able to plot your own way through a consultation, to be able to check in, keep something alive as a differential, or put something to bed as a differential. So knowing these diagnostic criteria is a real essential part of us, and that it is, yes, intuition comes into it, but you have to know the concrete basis on the, for these conditions as we go through. And there is now an array of these international and validated diagnostic classifications that we can draw on. Probably the big advantage or the big advance that we've had in the last 30 years, and it does go back to the original, be it Lewy or Alois Alzheimer's, or if you like, the forefathers of pathology and dementia. They all identified that there was misfolded proteins, but we probably become to understand this even more so. And of course, it's the type of protein and the area of the brain that's affected that results in the symptoms. And you've got to be thinking about both when you're having a look at it, and that's really that's the crux of a biomarker supported diagnosis. Is around pulling together some of the fluid biomarkers that we have, the imaging biomarkers that we have, but most importantly putting that into the context of the clinical symptoms. And Dr. O'Connor talked earlier on about atypical Alzheimer's disease, so you can just see with that one pathology how when different parts of the brain are affected, you can go from your typical symptoms of episodic memory loss to visuospatial symptoms if it's posterior cortical atrophy, to language predominant symptoms, as Lisa touched on, with logopenic variant Alzheimer's disease. So it's really key to kind of understand that. And it's also really, really key that we keep going back to these classifications. And I'm going to talk a lot about Alzheimer's disease because really that is the condition that we have progressed most at understanding, and it is going to shape our future approaches and is shaping our future approaches towards the other types of dementia. So while we aren't as progressed, it does give us a map as to what we can look and hope for the future in those other causes of dementia. For the majority of my working career, and up until 2011, and still in an awful lot of practices, this is the diagnostic criteria for Alzheimer's disease. I sometimes flippantly say is, if you couldn't think of anything better that it could be, it was Alzheimer's disease. But when we drill into it, It was only diagnosed on the basis that you had functional loss, so it was only diagnosed at a dementia stage. You had to have deficits or 2 or more cognitive domains, but that was in an era when it was very difficult to get access to neuropsychological testing. It still is sometimes. And there couldn't be any other obvious cause. And what we know from studies done over the last 25, 30 years, but even more recently with the amyloid PET studies over in the US, is that if you're just using this criteria, if you're just using your intuition, when it was done in the best of centres around the world, there was a misdiagnosis rate of about 30%, and that continues to be the case. So the importance of actually applying biomarkers is really important. And in the IDEAS study up in the US, they found that there was a change in practice in 60% of cases where amyloid PET was applied. So knowing and having, being thinking you're certain and being certain, or being able to fold in. So what that's led to is this evolution of how we diagnose from that era in 2011 when we started to fold in these research biomarkers that were coming through into the 2018 framework, which is kind of now the main framework that most of the subsequent classification for Alzheimer's disease are based off. And it's kind of led to this recognition that the pathologies, so amyloid, which is the main culprit that we see as the, if you like, the origin cause, or that most believe is the origin cause of Alzheimer's disease, followed then by tau phosphorylation, a few years later. But those pathologies start accumulating in the brain about 15 years before there's any symptoms evidence, and maybe even longer in some people. And that's led to what we call our ATN criteria, amyloid, tau, and neurodegeneration, or other biomarkers. And what we know is for amyloid pathology, we look at CSF changes in A beta 42, where we get a reduction in amyloid in the CSF because it's held in the brain. We get increases in our phospho-tau levels in the CSF. And we would do, here we're probably doing over 200 CSF biomarkers now a year. And that's kind of grown steadily since 2016, when we would have been doing very few. And then the other markers of neuronal neurodegeneration is what we talked a little bit about earlier on, where we look at atrophy patterns on MRI scans, and in particular in the medial temporal lobes, or elevations of total tau. So that's kind of where we're at. And really now that is because we have automated assays that we're able to measure that. That is, I think, standard of care, certainly for specialist services, certainly for people who are younger, and certainly for people who have atypical symptoms that you're interested in ruling out Alzheimer's disease. I think that's what should be an expectation for people to receive. But of course, really what this evolution in the diagnostic criteria has opened up is the space before dementia. So the identification of pathology without people having functional loss, and the opportunity then that comes to, if we disrupt that pathology, we can prevent the progression or delay the progression into those latter stages. So by the next iteration of classifications really folded in, first of all, prodromal, or what we call Alzheimer's disease mild cognitive impairment, where people have evidence of cognitive impairment but no functional loss. But also we're beginning to say, and I'll talk a little bit about some of those trials that Dr. O'Connor mentioned about pre-symptomatic populations, and our ability to identify pre-symptomatic populations. There was a little bit of a huzzah at the end of last year because essentially we had a break in the traditional Alliance on Alzheimer's disease classification. I'm sure you were all across it. It was very exciting. Ultimately, it came down to the Alzheimer's Association and the National Institute of Health used to actually get together and come out with a combined diagnostic criteria, and they kind of broke off. And they broke off on the basis of the Alzheimer's Society said that if you had one of your core pathologies present, even if you didn't have any cognitive symptoms, that was enough to diagnose Alzheimer's disease. So it was a big jump. The International Working Group, which are a group of clinicians, large group of clinicians from lots of different disciplines, have kind of countered it, and to my mind probably come up with a more reasonable interpretation on it, which has said that we still have to maintain it as a clinical biological construct, that we can't just go full yet to a pathological construct, and that there should be the presence of objective cognitive deficits along with the Alzheimer's disease biomarkers. And then the next thing is that the Alzheimer's Association criteria were just saying if we just remove the protein, that should be enough of an indication of efficacy, where the International Working Group have said that, well, yes, if you're going to have a therapy, it probably should, of course, help with the biomarkers, but there should be evidence of cognitive improvement. What was probably then, because the discussion was going on about the difference of opinion on reducing the diagnosis of Alzheimer's disease down to just whether or not you had amyloid, it was probably missed that they did actually incorporate and broaden out and bring in these new biomarkers, which I think is actually interesting. So as well as having the traditional amyloid and tau biomarkers, they introduced blood-based biomarkers, which I'll talk about for amyloid and for tau. They talked about tau PET, and then non-specifically they talked about neurofilament light. So these neurodegenerative and inflammatory biomarkers like GFAP, and then looking at non-Alzheimer's disease biomarkers, so the presence of other stuff that could rule out Alzheimer's disease or be indicative of co-pathologies such as alpha-synuclein and vascular changes on white matter changes on MRI. So really to kind of get to, what I think is going to be the main change that we're going to have to absorb over the next few years is blood-based biomarkers, and in particular phospho-tau 217. This original paper is from around 2021-22, when it became apparent that this biomarker in particular, was better than phospho-tau 181, which is what we measure traditionally in CSF, and the original kind of blood assays would have looked at, which wasn't as sensitive or specific as 217. 217 as a species of p-tau has come through as very, very sensitive. And in research cohorts, it was around trying to initially see how would it translate into real world practice. And really the accumulation of evidence over the last 3 or 4 years, 3 years really, and has been at an astonishing rate of its incorporation into clinical practice. Lumipulse, which is an automated, and that's the key thing. These will be blood tests that can be done in any laboratory within any hospital, okay, with the right equipment. It has excellent discrimination of amyloid PET scans, so those are the more expensive PET scans that are very difficult for many people to get, where you have the radioactive tracer that sticks to amyloid. So they correlate really, really strongly with it in clinical populations. It also correlates strongly with tau PET, which is another type of PET scan where it's the tau protein that we're looking at, and is often an important staging biomarker. But most importantly, when we look at autopsy, which is the gold standard, you see this huge, high, sensitivity, specificity with the, correlation with pathology, the amyloid and tau pathology that we see in brain after death. We ourselves, Adam Dyer, as part of some of the work he's done in our biobank here, has looked at validating within our clinical service. And the key, one of the key things was, first of all, it was very, very reliable, high sensitivity specificity, as had been in other populations, but actually the potential to avoid up to 60% of the lumbar punctures we do on the basis that you put in this two-stage where you put in a low bar through, under which it's probably definitely not, and then a bar above whereby if it's above, it definitely is, and then you're just dealing with those intermediate scores. And we know subsequently that if we add in things like GFAP or a beta 42 levels, we can actually reduce that intermediate zone even further again to higher degrees of accuracy. So the FDA have just approved it. It's submitted through that EU regulatory process, which will probably be quite fast. And it really is, as I've said here, is ready for prime time, because there is an abundance of literature and evidence that's coming out all the time. To the extent that at the recent Alzheimer's Association conference, they came up with a clinical practice guideline for its use. And I think there are a few things that we need to be very aware of. So they are not advocating for it as a screening tool. It should only be used in specialist memory centres with access to confirmatory testing. They were agnostic with regards whether it was 181, whether it was Lumipulse, whether it was Roche, whether it was, they really went on the basis of the pre-specified sensitivity specificity. So they kind of highlighted ones, certain species that could be used as a rule out, certain ones that could be used as a diagnostic tool, but the absolute consensus was it needs to be preceded by a comprehensive clinical evaluation and interpreted within consideration of the pre-test probability. Also to bear in mind co-pathology, and that they're not adjuncts or replacement for clinical assessment. And then, as I said, you can add in these other bloods to kind of tie down those intermediate cases. I'm not going to talk too much about tau PET imaging, other than people will maybe have heard of another imaging modality that other than the amyloid PET. It really hasn't kind of broken through yet to a clinical, to clinical services. What it is quite good at, it's very, very accurate. It does map to where we see tau species, but as we've heard throughout the day, you can have tau accumulation in an awful lot of different conditions. It remains kind of selective within the context really of a prognostic, and has been used in some of the clinical trials to do staging. So what we know is amyloid accumulation precedes tau protein accumulation, and as a result, people with lower volumes of tau are at earlier stages of disease, and that is useful in trying to discriminate between different populations. I kind of thought it was also important to highlight these are the anticipated or speculated curves for pathology accumulation and correlation with symptoms for frontotemporal dementia and for Lewy body. And it's the exact same process as we see with Alzheimer's disease, where there is this toxic protein accumulation preceding symptoms and different modalities and of biomarkers which will be useful. I wanted to touch briefly on neurofilament light in behavioural variant frontotemporal dementia in particular, and it was talked about a little bit earlier on. It's a neuron-specific protein that forms part of the neuronal cytoskeleton, and essentially it's a very reliable biomarker for neuronal injury, and we can measure it in the CSF and in the blood. And what we find is it is very, very sensitive, but not very specific. And if you look here in this paper, you can see that in amyotrophic lateral sclerosis, there's very high levels. There's high levels of it in Down syndrome, Alzheimer's disease. Higher levels then also in frontotemporal dementia. Levels aren't so high in Alzheimer's disease, but what it is a biomarker that essentially will say that there is a pathological process or a neuron pathological process going on within the brain. So where does it fit in? Well, neurodegenerative versus primary psychiatric. It is a well-validated blood test. It is available in some jurisdictions. It's not readily available for ourselves yet, but it may become available on our CSF panels. It's used, I suppose, primarily in the MS population for tracking response to therapies, but it will probably also fold over now into neurodegenerative areas where we look at it. And it can be used to distinguish between, let's say, people who develop primary psychiatric symptoms, to distinguish whether or not it might be an FTD or a behavioural variant FTD or a primary psychiatric. Also in this concept of phenocopy, frontotemporal dementia, which is a condition where we probably, and Emma Devaney from Sydney was over recently and she was talking about it, and it's a really interesting condition where it's probably people who had well-adjusted autistic traits, which when they get older, they become less able to manage their day-to-day with. And as a result, they present into clinics with a high degree of stress within the family, whereby they have more ritualistic behaviours and almost are presenting as somebody new with autistic traits. But actually what it is compensatory mechanisms are broken down. And they present very, very similar to somebody with the behavioural variant FTD, but when we do their FDG PET, we don't see the avidity, we don't see the natural progression. Huge amount of support has to go into families because there's a real struggle for them to understand what's going on, because they're being told, on one hand, they're hearing it's not dementia, and to a certain extent, they're almost gutted they don't receive a diagnosis. And actually, there's an awful lot of support that goes into trying to help somebody come to terms with the fact that it doesn't look like it's a neurodegenerative diagnosis. And in that instance, neurofilament light could be a very helpful biomarker to kind of indicate or rule out that there doesn't look like there's a neurodegenerative process. So it's not disease specific, but it's also highly prognostic. For helping to guide. And I just wanted to touch on, and Dr. O'Connor and Lisa were talking about the different, so frontotemporal dementia, that umbrella term that we take, but really it's such a complex interplay between the different pathologies and the different genetic drivers of some of these pathologies. And where we know, so behavioural variant FTDs, it's almost 50-50 between a tauopathy, TDP43, semantic variants, which are predominantly a TDP43 pathology, and then our non-fluent aphasias and things like that, which are often a tauopathy. And this is from John Hodges, which kind of just captures that interplay between understanding the different pathologies. And unfortunately, there are other biomarkers in development, but they're not quite there yet. So there's novel tau tracers to look at some of the conditions that Evelyn was talking about this morning. Novel tau tracers to try and distinguish between some of the atypical Parkinson's syndromes, seed amplification assays for TDP43, but they're not really there. And there is a new modality or a new methodology looking at extracellular vesicle tau and TDP43 as biomarkers in FTD and motor neuron. So I know Dr. O'Dowd talked about Lewy body pathology this morning, so I wasn't going to touch too much on it other than talking about CSF seed amplification assays, which are very close, but probably a few years away. And part of the challenge is the technology has gotten much better, but it's still probably a technology that is going to be limited, I would say, to probably most specialist centres or research laboratories rather than what we were talking about with the blood-based biomarkers for Alzheimer's disease earlier on. This was just it's a paper that's only out in the last few months, which looked at a two-step detection. And I don't know, did we talk about anosmia earlier on, or was it touched on? But I mean, just as a symptom that people can draw on, very, very sensitive, not very specific, but loss of smell, and how we kind of accurately measure for loss of smell. And to be honest, we don't necessarily do it brilliantly other than asking people in our own clinic, but it's hard to self-report. This was a study that looked at using the UPSIT, which is University of Pennsylvania instrument, and they used a two-step workflow whereby they looked at sense of smell, and then they followed it up by seed amplification assays for alpha-synuclein. And what they found was when you combine together a very, very high sensitivity and specificity for looking and for a diagnosis of Lewy body pathology after all, and it would have reduced the need for confirmatory CSFs in about 43%. So not something that's here yet, but I think a direction of travel that we're going to see for other conditions. And you can see how what we've done what's been done in Alzheimer's disease is following through now into other causes of dementia. Of course, the key thing is by biomarkers, our ability to diagnose earlier, our ability to diagnose more accurately, our ability to phenotype more accurately has meant that we're able to get better medicines to better groups of people. And really that era of disease modification where we've gone from really having medicines that worked on those very lower levels of the cascade to moving well up the cascade now and to try and interfere with it. It's sometimes confusing to people because they hear about amyloid therapies and they hear mixed messages. Some worked, some don't work. So what's going on? We know aducanumab ended up being quite controversial. There was one trial that was positive, one trial that was negative. The FDA approved it, the European Union didn't. But thankfully over the last few years we've had lecanemab and donanemab, two other agents that come true. And probably one of the key things to say is not all amyloid is the same. It exists as different types of aggregate within the brain. Some of it is very, very small amounts, and some of it is large clumped amounts together. And what we have known from a long time is the amount you clear and the speed at which you clear it is really, really important. And when we look at all of the different studies, we can see that the gantenerumab study, which was negative, really didn't get the amyloid levels low enough. We see that in one of the aducanumab studies, they didn't get low enough. The other one it did. And Clarity and donanemab Trailblazer study, where it got low enough, there was positive effects. Lecanemab was late 2023, and what we saw was a slowdown in cognitive decline in 27% of people. And donanemab was in May 2023, and similarly it was at 35%. And I'll talk a little bit around. So if we look at the efficacy outcomes, there was two different efficacy. There hasn't been a head-to-head. So this is pulling the data together ourselves. Okay, that there is a caveat when we do that. But what we see is, as a general takeaway, probably donanemab had marginally better efficacy, but it's very difficult to compare head-to-head because they didn't use CDR sum of boxes in both studies as the primary outcome. But marginally better efficacy outcome probably for donanemab. And when we look at the functional scales, what was key is donanemab also broke the groups down into people who had low or intermediate levels of tau. So it sectioned out people with earlier stage pathology versus people with more advanced stage pathology, which was the first time it was done in a clinical trial. And there is a clear signal in the Trailblazer donanemab study that treating earlier is better. And it was one of the first studies to do that. Lecanemab then also there was outcomes looking at quality of life, looking at caregiver burden, and it also showed that it was beneficial. Open label extension then just from this year's Alzheimer's Association. So we've 48 months now of follow-up for people on lecanemab, and what they've done is they don't have a placebo group to compare it to anymore because everybody's on agent, so they're comparing them to natural history cohorts that we have. And what we're seeing, or what is evident through the data as it's coming through, is there is a continued relative protection for people who are treated with lecanemab versus not treatment. And that seems to be amplifying over time as opposed to reducing over time. So yet very much strongly in favour of these therapies. What about the safety? Well, the main safety concern has been around these amyloid-related artifacts where you get little swellings or little bleeds in the brain. It's caused by virtue of the fact that these medicines disrupt the amyloid in the blood vessel wall and results in leakages or bleeds. Yet again, I've just kind of compared, and if we took the two kind of pivotal studies, Clarity and Trailblazer 2 that were presented, really you would say that lecanemab had a favourable profile with regards ARIAs compared to donanemab, in that there was a higher incidence of ARIAs in the donanemab program. What that led donanemab to do is to look at their dosing, and this was presented, and the FDA have just approved this, a modified titration protocol for how we give donanemab, and that has dramatically reduced the rates of ARIA to about the same as we would see with lecanemab in people. And it's not to kind of go into the deep or to go into the nitty-gritty with this. I just want to get a sense that this is, while we're not giving these agents in Ireland, thousands of people around the world are receiving these agents today. And the people who are attending our services are not getting to avail of them. And the science is moving on because they're in clinical practice and because clinicians are feeding into how we give them safer. And this is a really good example of how clinicians have worked to identify a safer means of giving these medicines. And the APOE4 genotypes were the group that were more likely to have these ARIAs, and they found that in the Trailblazer 6, they've also been had a positive response to that new titration. But this is a complex patient population, and this is a case of a 65-year-old lady in the Clarity study who actually had symptoms of an ARIA and received thrombolysis for her stroke, and unfortunately had a fatal hemorrhage. And there are implications for people who may be on these treatments for prolonged periods of time, and if they need thrombolysis or clot-busting medicines for the stroke or for their heart attack, that they need to know that they may not be able to do them. But we're not talking about a huge proportion of people. This is a study that we've done through our own centre here, where you're really looking at about 6-7%, and this has been replicated right across memory services in the US, in the UK, other parts of Europe, whereby you're looking at low percentages of people attending our memory services as we speak that would be having to avail of these agents. But the reason why we need to ramp up our services, the reason why we need to be doing biomarkers, is because this is really just a beachhead. These are not the definitive treatments. They're far from it. If this is where we stop, we'd be very disappointed. This is the program of agents that are in development, and what we have is multiple agents in phase 3 trials, the majority of which are looking at the biology of AD, the majority of which are focused on disease modification. So what to watch out for, this is my last slide, I think. Semaglutide is going to read out later on this year. Everybody knows that it's been the wonder drug. It's been trialled in Alzheimer's disease. There's been centres and many people in Ireland who have been involved in the EVOKE and EVOKE Plus studies. So we should get an indication as to whether or not that's useful. Prevention studies in preclinical Alzheimer's disease. So the use of lecanemab and donanemab in people with no symptoms whatsoever. And they should be reading out in both in late 2027. And combinations. So we have combinations of amyloid therapies and tau therapies that are in the DIAN-TU population and a few other populations where we're looking at thinking if we can take both of these, if we can target both of them, will we be able to get them out? And this is probably the compound that has caused most excitement, is a compound called trontinemab, which is a compound made by Roche. It's going into phase 3 study this year, and what it does is it's using a different mechanism for getting into the brain. It's using this transferrin receptor, and as a result it has very, very low rates of ARIA. And also, this was in data presented at AAIC, what we see is rapid reductions in A-beta. So by 106 days, somebody's brain is almost cleared. By 196 days, as opposed to like Clarity and donanemab, which were 18 months of treatment, we're looking at getting very, very extensive clearance of amyloid with very low risk of ARIA within 6 months of this therapy. And just wanted to finish out, still the most important study that was presented at AAIC was the Pointer study, which was a study that reported out on multi-domain lifestyle interventions. And it was a follow-up, really, I suppose, an extension of the Finger study. And it just emphasises that a brain health approach to dementia, and we haven't really chatted a huge amount about it today, but it is really, really important how we kind of incorporate, those lifestyle cardiovascular risk management, diet, exercise, and those kind of approaches, and that's really the paradigm to deliver. Yes, I'm very happy to talk about dementia trials and DRNI another time, and everybody is very much welcome to join those cohorts. So thanks very much.